Well, it's just come around to 1:30, and I think we should get started. My name's Chris Griffiths, previous president of the ESDR, and I'm delighted to introduce today's ESDR Kitchen event. For those of you who haven't tuned in before, these live webinars started in July of last year, and is our way from the ESDR to keep the scientific discourse continuing, and also to involve our members in academic and educational pursuits. We have 4 themes: the recipe book, which is new research techniques explained, which is the objective of today's session. Sweet and sours, when 2 protagonists debate a hot topic in the ESDR kitchen. In fact, everything takes place in the ESDR kitchen. Freshly baked is a discussion, usually from a couple of recently published papers by the lead authors. And then molecular cuisine is a senior distinguished individual from the world of dermatology research who will discuss his or her discovery pathways, or pathway usually, from research ideas to evolution. But we'll return to today's presentation, and flag the next slide. So this is from the recipe book, and it is from Beatrice Dyring-Andersen, who's going to discuss the Proteomic Skin Atlas. And this current session is sponsored by Sonkoly, but I'm going to hand over to my fellow board member, Enikö Sonkoly, who will introduce Beatrice appropriately. Thank you.
Yes, good afternoon to you all. My name is Enikö Sonkoly, and I will moderate this session together with Professor Julien Seneschal from Bordeaux. It is my great pleasure to introduce to you Beatrice Dyring-Andersen, who is going to reveal us the mysteries of proteomics in skin. Beatrice Dyring-Andersen is a physician scientist, and she has a translational dermatology research profile. She did her PhD at the University of Copenhagen in 2014. She was working on T cells, innate lymphoid cells in the skin, and how these changes arises. And then after her PhD, she did 2 postdocs actually. The first one she did with the NNF postdoc fellowship in Boston in Dr. Rachel Clark's laboratory, where she created a cohort of melanomas and investigated the immune system in melanoma. And that's where she met Professor Mathias Mann, who developed mass spectrometry, and they commenced a successful collaboration, which has resulted in the Skin Proteomic Atlas. And this is something we all think, I think, is very exciting. And now we will hear Beatrice introduce to us the Skin Proteomic Atlas. Please, Beatrice, share your screen.
Thank you, Enikö. You can see my screen now, right?
Yes.
Good. I would like to start by thanking the organizers for inviting me, and at the same time also thank ESDR for creating this wonderful platform where we can all learn from each other. I will talk about mass spectrometry-based proteomics in context of dermatology as part of the recipe book. And my experience for doing that is the Skin Atlas project that Enikö just mentioned, which commenced in 2016 while I was in Boston in Dr. Clark's lab. And that was a very supportive environment. And I contacted Mathias Mann, who's a famous physicist and doing mass spectrometry. And I asked if we could do mass spectromic analyses on disease skin. And he's told me kindly over a cup of coffee that I might want to start with healthy skin first. And so we did. And 4 years later, we published the Skin Atlas. And all the data is you are able to acquire and is and can be downloaded on skin.science. But this is the background for me talking about mass spectrometry based proteomics. And so I won't discuss the Skin Atlas project as such, but I will say that we cannot discuss all the dimensions of the proteome in 20 minutes. So I will do an introduction on label-free quantification using LC-MS. And first, I'll introduce proteomics as such, and we'll talk about what relevant samples that are useful in context of dermatology when doing mass spectrometry analyses, and talk about the mass spectrometer itself and label-free quantification. And the last 3 slides are about making sense of the data, but I won't go in too much into the data analyses. That has its own webinar. So the proteome is, as you intuitively would think, that's the entire set of proteins expressed by tissues or cells or an organism at a certain time. And the proteomics includes what we, of course, the identification and quantification of those proteins, but it also includes the localizations, modifications, and interactions, which ultimately is their function. And before I started this, I was used to doing immunohistochemistry, for example, as you can see in the section there. And one of those sections could take a couple of days. And with mass spectrometry, we were able to do thousands of proteins from one sample. Now, the caterpillar and the butterfly, they have the same genome. And what's interesting is how that genome is regulated. And that is what makes the difference between the caterpillar and the butterfly. And it's the same thing with humans. Now, I don't have a photo of the same person with 80 years apart. So this is my son 10 years ago and my grandfather. But the regulation is the same principle applies, that we have the proteome, which is the machinery and the end stage. And I show that with this slide because there you can see that we have the genome made up of 4 letters combination that makes this code for what is us. And then that's transcribed into RNA. And the RNA itself, there are variation and regulations of that, which makes it a bit more complex. And in the next step, which is the translation to proteins, we have proteins made up of 20 amino acids. And those can be modified by themselves also. So we have this immensely complex system called the proteome. Now, even if it is immensely complex, we still like to work with it. And in dermatology, we're so lucky because we have this very nice tradition of translational research, which means that a lot of methods have been developed for us, and we have an accessible organ to work with where we can take biopsies. And biopsies is maybe the most prevalent sample to work with. And both fresh tissue and formalin fixed and paraffin embedded tissue are useful when doing mass spectrometry analyses. They have some challenges, and I'll come into that, but both are useful. Now, punch biopsy, you don't need more than a 2 millimeter biopsy to get a lot of material to work with. And with the blocks, with the paraffin blocks, a couple of sections at the most, and you also have enough to work with. The sections can be combined with laser capture microdissection, and this is a field that's moving very fast. And today we're able to cut out cells of interest and get a fairly good proteome out of that, of thousands of proteins just from a couple of hundred of cells. Tape stripping is, there is a lot of literature on that, and I thought it would be challenging to work with, because when you take a tape strip, takes parts of stratum corneum, and that's what it can do. It doesn't go deeper. But in stratum corneum, you have a lot of structural proteins, and those are a challenge when you work with mass spectrometry. But it turns out that we can get approximately or between 1,000 to 2,000 proteins from tape strips. And flow cytometry is also very useful. You can sort the cells you have of interest from the skin or from the blood, and then downstream use mass spectrometry. Now, this is the challenge when working with biopsies of skin, because the skin is made such so it's resilient and it has this toughness. And that is made by structural proteins such as lots of collagens, keratins, elastin. And to work with that, you have to break them down. Otherwise, we don't get a representative proteome. And for that, we use lysis and sonification of the proteins, of the samples. And this process leads to the disruption of cells and tissues, and we solubilize the proteins. And in the mixture, we then have these proteins that are folded. And to work with the label-free quantification, we need peptides, parts of proteins. And to get that, we need to unfold the proteins and then use proteases to cut them into smaller pieces. And so for that, we use chemical reactions. The first one is reduction, which will unfold the proteins, and alkylation to prevent the process of the binding of the peptides, and so they don't form disulfide bridges again. And that leads to the last phase where you can then use proteases, because the protease will be able to come into contact with the whole proteins. And that will give us the peptides. Now, why would we use proteases? It's called, what we're doing is bottom-up proteomics, which means that we identify proteins by looking at peptides. And the only reason we can do that is because some peptides over a certain size will be unique for a protein. And so we use proteases to cut the proteins in smaller peptides. These are easier to dissolve and ionize. I'll be back to the ionization process. And that gives us a better chance of identifying them at peptide level and then protein level as well. Now, the most commonly used proteases are trypsin and lysine, but there are other proteases that can be used. And for trypsin, it cleaves specifically at arginine and lysine, but not when it's followed by proline. And so we get this ideal average size of peptides that is good for the mass spectrometric analyses. And this is how they can look. And these are the ones we made the skin atlas data on. First, we use liquid chromatography. And the machine itself is called high performance liquid chromatographer. And what it can do, it can separate the peptides based on its phases. So it uses the chemical properties, or we use the chemical properties of peptides to separate them from each other. And so when they are eluted from the liquid chromatographer, we have a less complex sample. And that's important for the next part, because the peptides are ionized, and that process is called electrospray ionization. And John Fan got the Nobel Prize for that. But it means that we ionize the peptides in small droplets containing solvated ions. And that goes into to the mass spectrometer and is analyzed. Let's see here. And what we get out of that is a record of mass spectrum abundance versus mass to charge. So the peptides will hit let me see, the ion detector, and from that we'll get a spectrum. Now the mass spectrometer also fragments the peptides, so we get an even better quantification by having two sets of identification, one of the parent ion and then the fragments. Another way of making the samples less complex is by using peptide fractionation. And that means that we take one sample, which is our soup of peptides, and we elute it through the liquid chromatographer offline. So it's not, it doesn't use mass spec at that point. And we can make one sample into 8 or 16 or 48 samples, depending how much we want to fractionate the samples. And it's using a pH gradient to divide the peptides. And so that way we can have a lot more samples from just one and have the possibility of getting deep libraries. And so once we have one sample becomes 8 or 16, and then next when it goes through the HPLC and the mass spectrometer, we have a lot more elution or material to work with and less complex samples, and we get more hits. And this is how we can look. So if we look at B here, this is a single shot from a sample. And all the peaks there are peptides, and this is time. And we can see that they are eluted when we do it as a single shot of the whole sample. If we instead use fractions, and the number here is which fraction it is, so we see that fraction 3 comes out in maybe the first 10-15 minutes of dilution. The next one comes out maybe 20 to 40 and so on. So that's a way to divide the samples and get better data. And for the last part, that's the data analysis, which is pretty challenging as well, because how do we make sense of these spectra that we get? And is it good enough, the data that we acquire through the mass spectrometer? Now, for the Skin Atlas project, we prepared libraries of the skin layers and of the cells. And that way we were able to identify 10,701 proteins. And we used the libraries to match our single shot prepared samples to our library. This can also be done without a library, but then you match it to a database directly. And so for now, or when I've tried it, I get less hits. And I think that with library preparation, you are more certain of the data that you get. Now, for doing the data analysis, which I don't think I'll get into that, but you can always ask questions about it if you have. It depends on how you have done the data acquisition. So for some of it, you can use MaxQuant to identify the proteins, and that is for data-dependent acquisition, it's called, or Spectronaut for data-independent acquisition. And for the bioinformatic process downstream from there, I have used Perseus, and but there are other software out there that can be used for this. One of the newest software that's been made by a good colleague of mine, his name is Alberto Santos, is the clinical knowledge graph. And this integrates the data and is made so it becomes more intuitively and easier to work with this data. Now, to talk a little about the challenges with mass spec data is the protein abundance, which means that the great dynamic range that you get from the data. And I'll come back to that in the next slide. Another challenge is that some of the peptides might not be in the database. And lastly, if we don't measure, and we know that we're not measuring all peptides when we do mass spectrometry analyses. And so if we don't find a peptide, we don't know if it's either not there or very low abundant. Now, the advantages are that it's a fairly robust system and that we have a reliable software for analyses. And we also have roots quite well understood. So if I show you now the dynamic range problem, so here we have the intensity. It's a log 10 scale, and we have the intensity of proteins. So up here is the highest abundance proteins in the outer epidermis, and this is keratin 1 and 14. And down here we have all the proteins that were identified in the skin atlas, 10,701. So you can see that there is a 7 orders of magnitude between the highest and the lowest abundant proteins, which is very challenging to work with. And so when we work with the data, it necessitates that we do a lot of quality controls. We normalize our data. We have to think about how to do imputation, although I won't come into that in details, and then applying statistic that makes biological sense. And now I'll show you a couple of examples from the skin atlas. Now this is a dynamic range plot again, and we have the intensity here and the proteins as I showed you before down here. And you can see that it's, as a quantitative measure, it's pretty good because we have collagen 3 here, which is pretty abundant in the dermis, as we can see in the immunohistochemical photo, and collagen 4, which is less abundant. But we can identify proteins that are so low abundant that it doesn't even make sense to we can't do stainings to validate those findings. Now here is one for the melanocytes where we see melan A, which is a melanocyte-specific protein. We know that the melanocytes are in the basal layer of the epidermis. And here we see what the mass spectrometry analysis finds. It finds it in the melanocytes and in the inner epidermis where it should be. And this is a principal component analysis. And I know that Tiago Matos showed you these in his talk, but this is to show you that it's a very robust system. I mean, I didn't expect it to be so detailed that we could see differences between dermal and epidermal T cells, for example. And you can see that all the cells, the different cells, they cluster together in groups. And that for me shows me that it's a very robust and good system. Now for the last part here, I've assembled a couple of resources that I use. The string database, where you can acquire proteins and it shows you the networks of them. Protein Atlas, where you can find photos of pathology and you can find the gene expression levels, protein expression levels. We can query data through Enricher as well. And then, as I mentioned, the clinical knowledge graph. And then we have created a repository for the skin atlas data as well, if anyone wants to continue working with that. And that brings me to my last slide, which is the acknowledgement slide, because I would like to thank you for your attention and all these people on the slides that are my collaborators and dear friends and always help me. Thank you so much. I'm happy to take any questions.
Thank you very much, Beatrice, for this really brilliant talk. It was very clear to get an overview on this proteomic analysis. So you can submit your question through the chat, and we have a question for you. It's related to the peptides, for example, antimicrobial peptides that are already present on the skin at the time of sampling. So the question is, are they always present throughout the sample preparations until acquisitions, or are they excluded during fractionization?
Are you thinking if they are, so if it's an artifact because we do something with the skin and then we get their expression? Is that the question?
Yeah, probably if for these peptides, can they probably be modified during the different step of the sample preparation, something like that?
Yeah, so that's what we also, when we thought about that, when we took the samples as well, and we try to work with it so we don't affect the tissue too much. When we got the skin, we picked it up ourselves and made sure that we worked on it as fast as we could. But there are a lot of changes that occur very fast, right? And especially post-translational modifications. But we're not looking at those. So I think we got a clear representation of what's there, but of course in the details and post-translational modification, those can change, yes.
Yes, thank you very much, Beatrice, for the beautiful presentation. And I have also some questions actually. So one question is, you said that you find some peptides which are not in databases, and as you know, there are, or maybe know that there are some long non-coding RNAs that are predicted to code for maybe some micropeptides, and this is something pretty new that has been found lately. And is it possible in your mass spec data to search for these peptide sequences, or do you just throw away these peptides which are not in the databases?
As long as they are in UniProt, we get some kind of name for them, right, or some numbers, but not.
I rather mean if they are not, but you only have the sequence, the predicted sequence, just as you do BLAST for RNAs or
so
DNA sequences.
The FASTA file that we use also has predicted proteins, but I'm not sure it has the ones you are talking about. So if they're not in the FASTA file, then it won't come out in the data as something we can use.
I see. Okay.
Good. Just another question. Of course, you show data on the human normal skin, right? So of course, do you have the chance to look at inflammatory condition? Because I think there is a ton of peptides that are not seen in normal condition.
Yeah, there are huge differences. I mean, for example, in just fibroblast in psoriasis, which is not a fibroblast disease, right? They are totally different, and so I think that there is a huge potential to identify also like clinical phenotypes, right? That we don't really understand, and I think there is a potential to use mass spectrometry on that.
Yeah, there are a lot of questions to you, Beatrice. Now some technical ones. So what should be the minimum quantity of cells collected by laser capture microdissection for proteomic analysis?
In whose hands? I mean, we get reasonable data on 300, but I mean, maybe someone else can get more. And what is reasonable, right? Is it we get a couple of thousands of proteins on that?
Yeah, I guess it's difficult to say, but you work with what you have. And there is also another question from the same person. Then, have you involved a hair follicle proteome in the Skin Atlas project?
No, we haven't. And that, but I mean, we could have pulled hairs, right, and taken some. If you get the follicle there, we could have investigated that. But I think to do that, I would rather use laser capture microdissection, and then you can get the different parts, right? Or you can stain the cells that you want to look at and do it, yeah, separately.
And we have another question coming from Toulouse, close to my city in Bordeaux, France, from Michel Simon. You talk about data normalization. So the question is, if you would like to compare two samples, for example, what do you use for that?
Okay, so the simple answer would be that you can do log2 median subtraction and then filter for example, 70%. Okay, but if there is also the possibility of normalizing to the size of the protein. Because if you have a protein with a lot of peptides, that would be more, you have a higher risk of finding that one. So that's a way to normalize as well. But the simple answer would be to normalize by log2 median subtract and then filter.
Yes, thank you very much. I think that time is up. There are a couple of more questions, but maybe you can answer them afterwards.
Okay.
Thank you very much for your very nice presentation.
Thank you.
And thank you all who sent questions.
Okay, well, thank you so much, Beatrice. That was a very well attended session as well. And thank you for answering the questions so well. And as Enikö and Julien have said, there are a lot more questions coming in, which I guess we can probably answer later on, maybe by email. So we'll move on to what's going to happen in the future. Well, actually, we're going to give you a day off after Easter. So there's no ESDR Kitchen webinar on the 7th of April. So you'll have to do your own cooking on that day. But on the 21st of April, we're all back in the kitchen for our freshly baked session, which will be 2 newly published papers, and the lead authors are Trong San Fan from Germany and Min Ma from Switzerland. I'm looking forward to that kitchen session. And just as we close, just to remind you that the abstract submission deadline for the ESDR meeting this year is the 31st of May. So hopefully you have been able to continue with some research and have research that you'd like to present at our virtual ESDR meeting in September. So thank you so much for joining us today. Thank you again, Beatrice, and look forward to seeing you in about a month's time. Okay, goodbye.